So, if we look at a cell membrane in the human body, whether it be any cell, whether it be the neurons or any other cell, then we can find out that the concentration of sodium ions, the concentration of sodium ions is much more outside the cell as compared to the inside. Now, this causes the overall membrane potential to be a negative membrane potential. So the outside is more positive and the inside is comparatively negative. Although the positive ions are there outside and the positive ion is there inside, but comparatively the inside is more negative than the outside. And this membrane potential is called the resting membrane potential. Resting membrane potential. And this resting membrane potential is normally negative 70 millivolts. So this is the voltage at which normally the cells reside. Now what happens when there is a nerve impulse? Let's say this is the axon bulb of a presynaptic neuron and this is the postsynaptic neuron membrane. Now this postsynaptic neuron membrane would contain some gated channels which are chemically gated channels, and these are channels for sodium ions. Now when the presynaptic neuron, it releases a neurotransmitter, then this neurotransmitter binds to this gated channel and opens it up, as a result of which the sodium moves inside, the sodium ions move inside the cell. Now you can see if the sodium ions, they move inside the cell, then the membrane potential would change. So if we plot this whole thing in a graph, let's say this is the y-axis and this is the x-axis, and we are going to plot the y-axis to be the voltage in millivolts and the x-axis to be the time. So let's say this is 0 millivolts and our resting membrane potential is somewhere around here, which is negative 70. So like I said, when the sodium ions gated channel, they open, the sodium rushes inside the cell, rushes inside the postsynaptic neuron. And this causes the membrane potential to start increase. Now, as the membrane potential it starts to increase, it goes to about positive 30. It goes to about positive 30, and at this positive 30, some other channels, which are the potassium channels, they open. So this potassium channels, these are voltage gated potassium channels. As the voltage is increasing and reaches positive 30, this sort of opens this potassium channels, this K plus channels. Now on contrary to sodium ions, potassium ions are more present inside the cell rather than being outside. So as the potassium channels, they open, the potassium ions from inside the cell, they rush outside the cell. Now as the potassium channel opens and the potassium ions, they rush out, what happens? The membrane potential again starts to restore. So it again gets to negative. Now one thing, these potassium channels, they can open very fast at positive 30, but their closing is very slow. So they need a lot of time to close, or they're very slow closing. So because they are slow closing, when the membrane potential it reaches to about -70, the potassium channels, they cannot close, and this sort of skids to about -90. And eventually, as all the potassium channels, they close up, and again the membrane potential, or the resting membrane potential, is restored. So first, let's go again. The -70 is the resting membrane potential, and as the sodium ions, they move inside the cell, the -70 resting membrane potential, it starts to go towards the positive, and we call this phase the depolarization phase. The depolarization phase. Now, as it moves towards +30 and the potassium channels open, because the potassium channels, like I said, these are voltage gated potassium channels. They respond to certain voltage. Change in certain voltage. So the potassium channels open and the potassium starts to rush out. And again, the membrane potential, it comes down, and we call this phase the repolarization phase. Repolarization, because it is again getting polarized back to the resting membrane potential. So repolarization. Now, when the repolarization comes to about -70, it should have stopped over here, but it skids out and goes a little bit more negative. And this skidding is called the hyperpolarization. So after the hyperpolarization, as the potassium channels, they eventually close, the resting membrane potential is again achieved, and it comes back to -70, and the action potential ends. So this was the overview of the neuron action potential. So that was it for this video. If you like it, then give it a thumbs up and share it with someone who would need to watch it. And before going, you can check out my other videos, which are going to be linked over here and also in the description down below. And until the next time.